Electron microscopy. In electron microscopy, electrons are propelled by a charge of 50,000 volts and the illumination of focused by electromagnetic lenses instead of conventional glass lenses. The electromagnetic lenses means that electron beam is allowed to pass through an electromagnetic field created by electromagnets. It does the same job of converging the beam as the ordinary glass lens does in the light microscope. Because of such high resolving power of an electron microscope and because the subcellular particle lies much below the level of resolution by a light microscope, the electron microscope has become an indispensable equipment for cytological studies. The electron microscope was first built by Ernst Ruska of West Berlin, who shared 1986 Nobel Prize in Physics with Binning and Roher. Preparation of specimen for electron microscopy: fixation, sectioning, staining, shadowing, negative staining, etc. Since the specimen examined in an electron microscope is subjected to very high vacuum, it cannot be examined in living state. Further, a technique for fixing, sectioning and staining used for light microscopy are not suited to electron microscopy. The material is first fixed with glyceraldehyde and then with osmium tetroxide, the former cross-linking the proteins with their neighbors and the latter binding and stabilizing the lipid bilayers and proteins. The specimens are cut into very thin sections, about 1/200 of the cell thickness, since the electrons have very limited penetrating power. This is made possible by dehydration followed by permeation of material with a monomeric resin, giving a solid block of plastic which is cut with a glass or diamond knife on an ultramicrotome. The thin sections obtained are placed on a small circular metal grid for viewing in the microscope. In electron microscope, the contrast depends on atomic number of molecules in the specimen. Since most specimens have carbon, oxygen, nitrogen and hydrogen, each having a small atomic number, they are impregnated with salt or heavy metals like uranium and lead with high atomic number. The degree of contrast depends on the degree of impregnation or staining. Sometimes for electron microscopy, the specimen after fixation and drying is sprayed with the heavy metal which make a thin film. The metal is sprayed with an oblique angle so that the coating is thicker in some regions that in other. The process is called shadowing because it creates a shadow effect, thus increasing the contrast. For negative staining, macromolecules like DNA and proteins supported on a thin film of carbon are washed with concentrated solution of salt of heavy metal, uranyl acetate. After drying, the specimen will have a coating of salt of heavy metal covering the carbon film everywhere except there where macromolecules are present. Since electrons will pass through the macromolecules more readily than other places having a coat of metal side, this is described as negative staining, which give a negative image of the specimen. Transmission electron microscopy versus scanning electron microscopy. The transmission electron microscopy can be used the study of surface of a specimen. For this purpose, a thin film of heavy metal is evaporated on the dried specimen from an oblique angle so that the coating is deposited, which is thicker in some places than in other, a process called shadowing. Due to shadow effect, the image give a three-dimensional appearance. This can be satisfactorily done for thin or small specimen. In contrast to transmission electron microscopy, where electrons pass through the specimen to form an image, in scanning electron microscope, the electrons are scattered or emitted from the surface of the specimen. The specimen is fixed, dried and coated with a thin layer of heavy metal. This specimen is then scanned with a very narrow beam of electrons. The quantity of electrons scattered or emitted from each successive point on the metallic surface is measured and used to control the intensity of second beam. which move in synchrony with the primary beam and forms a highly enlarged image on television screen. The SEM technique provides great depth of focus, and because the quantity of electrons scattered depend on the angle of the surface of the beam, the image gives a three-dimensional appearance. Thank you.